Well, thanks, Mark, for the introduction. Thanks, Norberto, for the invitation. I am very honored to present, to come back home. And thanks for our colleagues from the lab to working so hard to putting up such an event. So it's unbelievable because a lab can organize such an event. It's quite an accomplishment. So the nervous and immune systems, signals and connections are becoming a great subject of scientific inquiry in the last few years. So they are kind of becoming intertwined systems. Nervous and immune systems, they share a lot of similarities. They are based on the secretion of small signaling molecules, so neurotransmitters and cytokines. They have surface receptors on the target cells that mediate the effects of these small molecules. They are based on information flow, so the immune system is basically keeping the self separated from the pathogens and strange bodies that can invade your body. And the nervous system is also kind of making information processing from the outside world and also keeping track of the whole qualities of your inside body. So it keeps also tracks of who is yourself, who is other else, and are other people, and communicating. So they are you can think of these systems as information processing complex systems. So we came to the complexity that is a theme of this roundtable this morning. So why they are complex systems? Well, it's not because you take years to learn how they work, and have heavy books explaining each one of them. Actually, they are complex systems because they are made of many, many, many different parts. And the interaction of these parts creates emerging properties. So you have properties that arise from the interaction of these different parts of both nervous and immune systems. So these complex behaviors of these systems, you cannot tell just by examining the behavior of just one of these parts. You have to take the whole system into account. And that's why it's so complex for us to understand these systems. So we are reaching a point where the connectome is maybe the use of brute force to map the connections of the nervous system. I don't know, the synapses, or maybe the whole cascades of signaling molecules in the nervous systems. And is an approach that sometimes I will put an interrogation point on the connectome and thinking maybe is a big science, big data approach. I will not agree that everyone has to think in this way. I still think there is a wave of genome, how's the genomics and all this proteomics stuff. We have to be careful of not following fashion, in science. So besides the similarities between these two systems, we have also some differences, of course. I would point to, actually I was thinking about the differences and it was really hard for me to pinpoint the differences. Anyway, we have a centralized nervous system, especially in tetrapods. It's very important that we have a centralized nervous system versus scattered immune cells that works in hubs in our body. Of course, you have this ganglia and you have agglomeration of immune cells, but they work more as a scattered network in close contact with every reach of the body, in the contrary of the centralization of the nervous system that is centralized and, of course, has reached whole points of the body, but has clearly a centralized hierarchical system with the central nervous system on top of everything. And the other big difference is that you have the specificity versus pleiotropy. So you have a specificity between neurotransmitter and its receptor, and in the case of the cytokines, what you have is that one given cytokine can interact with many different receptors. So it's easier to keep track of the receptors than keeping track of the cytokines. And does it make sense to have like we say, a glutamatergic system of transmission? It doesn't have a CCL system of transmission of information. So we have to keep track of the receptors. So this brings us for questions to solve between these two systems. So Number one, there are cytokine secretion receptors in the central nervous system. That will be the first question. And this question has been answered actually just in the last 20 years. So number two, what are cytokines doing in the brain if they are there? Number three, are cytokines neuromodulators? And number four, are they relevant in neuropathologies if they are neuromodulators? And fifth, can they be therapeutical targets if they are neuromodulators? So they play a relevant role in any neuropathology. I hope I can show this until the end of the talk. And six, if they can become therapeutical targets, is it economically feasible to do this? So would be worth to put the money on this kind of treatments? I hope I can show you also answer this question to the end of the talk. So yeah, I will give a straightforward answer to the first question. Cytokine receptors and secretion occurs in the central nervous system, and they are mainly in these regions, in the hypothalamus, the nucleus accumbens, hippocampus, thalamus, neocortical areas, and the cerebellum. Mainly in these areas. And I'm going to give special relevance to the hippocampus because of the nature of the meeting. So yeah, we can say that we can put like a green color here and say, yes, of course, there are cytokine secretion receptors in the central nervous system. I'm going to show some pictures later on so you can believe me. So this brings us for our second question. So what are cytokines doing in the brain? So if they are secreted in the central nervous system, you have receptors in glial cells and also in neurons, what they are doing, what's going on in the brain. So this is more tricky question to answer, and I'm going to start refreshing your memory, or for the ones that are not familiar with, that cytokines are classified in families. So first discovered were the interleukins because they were exchanged between leukocytes. You have the tumor necrosis factor family, the interferons family, and the chemokines. Chemokines are special cytokines. They have chemoattractant properties to cells of the immune system. So I'm going to give special attention to chemokines in this talk because they are relevant for the epilepsy. So chemokines are classified by cysteine residues that are present on the N-terminal of the protein. So you can see here in this picture from Corridae that you have one cysteine is a C chemokine, you have two cysteines side by side is a CC chemokine. You have one residue in the middle of these cysteines is CXC, and you have also the family of where you have three residues between the cysteines, so it's CX3C. It's a kind of tricky nomenclature, but it's good. It works like, you have the structural nomenclature and the functional also. It's kind of molecular biology works this way. You have tons of letters to play with. So now I show you how the pleiotropy works. So you have listed here the receptors, and you can see that each receptor can interact with many ligands, and I will pay attention especially for this CXCR4 receptor right here. He connects with SDF1, so the ligand is CXCL12. Should be, all right. So you can see that's a big difference from the neurotransmitter receptors, where you normally you have a ligand and a family of receptors, maybe metabotropic and ionotropic receptors. So this kind, they are all G protein coupled receptors, and they can be affected by many different ligands. Actually, this picture, this list is very old for cytokine standards. It's 10 years old, so I was checking the new lists, and it's quite limited in his. We have CXCR7, and actually CXL12 connects to these two receptors. So you use it to believe there was just one ligand, one receptor. That's quite rare for cytokines, but now we know there's two receptors for the same ligand. So CXCL12, like I was telling you, and CXCR4, they are very interesting for us because they are expressed in hippocampus during development, and they keep being expressed by special cells, the Cajal-Rizzo cells in the hippocampus, and I will show later on they play an interesting role in the hippocampus. So they are necessary for the migration and organization of the cells during development. And in the adult animal, you have less expression. So this is like embryonic animals, and you have P0 here in the lower panel. So we have here, this was a work made for our group in the Texas A&M University and my postdoc in Dr. Shapiro's lab. And this is the pyramidal layer of CA1. And you can see here the CCR2 receptor in neurons, and this is a naive animal. So the receptor for this chemokine is present in the hippocampus, in pyramidal neurons of naive animals. So here you can see that astrocytes, and these are astrocytes in the hilus of the hippocampus, they also express receptors for the interleukin-1 beta. So IL-1R1, you have 2 receptors for this interleukin. So yes, receptors are expressed by neurons, astrocytes, but we are still trying to figure out exactly what are those receptors doing in neurons. So we have this publication by a group, Marchioni and others, where they have transgenic mice that expressed enhanced GFP protein with controlled by the tagged in the gene for CXCR4. And you can see here the neurons. They have the Cajal-Ritzius neurons in the neocortex. They disappear early on the development, but they play a big role on organizing the structure of the neocortex. And in the hippocampus, those animals are a month old, and they still keep the Cajal-Ritzius neurons in the lacunosum molecularis. So this is the entrance point of information for the dentate gyrus. So here is the molecular layer, and here are the granular cells of the dentate gyrus. So these cells are positioned in the entrance point of the flow of information in the dentate gyrus. And they have shown that these neurons in electrophysiology studies, here is a control of the membrane potentials here. And if you apply in the BEF, the CX, this is another name, so STF1 stands for the CXCL12. If you apply this in the BEF, the cell hyperpolarizes. So it has a direct electrophysiological effect on these neurons. So I will tell that one hypothesis that was formulated for the cytokine's role during the immune influence on the nervous system functioning, the most impressive results are about the sickness behavior and depression, because sickness behavior is that every one of us have this experience in our life. When you get the cold and you get the flu, and you are like feeling really bad and really like not wanting to talk to anyone, you want to go to bed, you are lethargic, you are like hypothermic. And what we have here is that you have this infection, you have the peripheral infection, and you have great increased levels of TNF and IL-1 beta. And what was the big discovery was that this behavior were not because people were out of energy or distressed because they are sick. Actually, what was going on is that this behavior is a functional way of the body to protect itself. So the immune system seizes control of the nervous system and induces a complex behavior in humans. So this was discovered, this was confirmed especially because when cancer patients underwent treatment with IL-2 and interferon alpha, 30% of these patients experienced a major depression event. And this was a great surprise because, of course, they also thought, okay, these people are under cancer treatment, so they have a major depression episode because of the disease. But for controlling studies of placebo, later on, they proved that no, actually the effect was because of the interferon and the interleukin. So depression, and I recommend this as a must-read for anyone who is interested in the immune modulation. This review was published in 2008 by Robert Dancer. So it is exactly like this, from inflammation to sickness and depression, when the immune system subjugates the brain. I like the title, of course. So the second question is like, I will keep this as a read, because what are cytokines doing in the brain is still, for me, is a very open question. We have barely scratched the surface of what's going on between these two complex systems. So I'll keep this like well, we barely know what's going on. So let's keep this in red, and let's think like, okay, so cytokines and receptors are present in neurons, in astrocytes, in glial cells, in microglia. They play electrophysiological roles. They are well located in regions. They are important for learning and memory. So are cytokines neuromodulators? This is our first question. This is our next question. So I would say that the area that has more information is exactly one that's very important for people who works with temporal lobe epilepsy, because the main system of learning and memory of the brain, that is the LTP going on neurons, on special synapses in the hippocampus, is where neuroplasticity occurs, and you have all this learning and neurogenesis is necessary for keeping this process. So this whole reverberating system depends on IL-1 beta to occur. So you have peaks of IL-1 beta secretion, and you have IL-1 beta secretion during development, and that's very interesting that IL-1 beta still is expressed in high levels and is necessary for LTP. So if you block IL-1 beta with the endogenous blocker, there is IL-1RA, is amnesic. So, and if you have a knockout mice for the IL-1 beta expression, these animals has trouble in learning new tasks. And if you recover this animal giving IL-1 beta, it recovers the memory formation on memory tasks. So it's interesting because IL-1 beta plays a role in plasticity and building of synaptic circuits in the whole brain during development, and it keeps this role in the hippocampus, because the hippocampus is exactly the great plastic area in adult animals, so it makes sense. But was a great surprise that IL-1 beta was so important for this mechanism, right? So it helps phosphorylate the NR2 subunit of the NMDA receptors. That's why it has such an influence on LTP, and also controls the tryptophan, also controls neurogenesis through the control of the tryptophan kynurenine pathway. So long-term potentiation is also modulated not only by IL-1 beta, but also by TNF and IL-6. TNF is a very interesting cytokine because it has two receptors, and they have opposite chains of secondary messengers. So sometimes it can blocks LTP, sometimes it can enhances LTP. And IL-6 also normally has a block role, so is amnesic when you have large quantities of IL-6 in the hippocampus. So the third question, I would say, are cytokines neuromodulators? Yes, for sure, they are neuromodulators. They are like neuropeptides, and there are some hypotheses that say that the chemokine system in the nervous system would be the third major communication system besides the neurotransmitters, neuropeptides, and then you have chemokines. You have cytokines in the central nervous system. So then it leads us to the fourth question. So are they relevant in neuropathology? So if they are neuromodulators, which role they play on neuropathologies? So our big interest is in epilepsy, and when studies measuring cytokines in serum from epileptic patients, they could measure highly elevated levels of IL-6, TNF, IL-2, IL-4, interferon gamma, and IL-1 beta. And CSF after seizures was collected from spine tap in patients, and was elevated levels of IL-6 indicated maybe a role for this cytokine in the epileptogenesis, but still. So in temporal cortex from patients with familial epilepsy, they have elevated levels of IL-6 and CCL2. And those are results from Singha from India and Choi from Korea. And a recent paper was published with resected tissue from TLA patients. And what they found was this group from Holland, Kahn, and co-workers, they found that temporal lobe epilepsy plus hippocampal sclerosis, they have elevated levels of IL-10, interferon gamma, and ICAM-1. And this was interesting because they used a multiplex analysis, so they made a huge panel with dozens of cytokines, and that's the correct way of doing, like you do a shotgun and you measure the whole cytokines, and then you figure out by principal component analysis and other kind of sophisticated and statistical analysis, and you can figure out what's going on with the cytokines. And we still don't know what we still don't know is about the receptors and about which receptors are activated. So this will be the next steps for further research. But anyway, they found interesting patterns. Actually, there are more, like 5 different patterns. I'm just showing here the TLE, the temporal lobe epilepsy, and these were elevated in the hippocampus and not in neocortical areas. So on the TLE patients without the hippocampus sclerosis, you have elevated levels of CCL2 and CCL4. So here is it's a work from our group. So now I'm changing to experimental animals. So these are pilocarpine animals, 5 days after status epilepticus. And what we have observed in the hippocampus was an increased presence of both the receptor and the CCL2, the chemokine, the ligand for this receptor. So here we have the stereology from the number of CCR2 positive cells in the hippocampus, and there was a statistical significant difference in CA1, and the CCL2 protein was largely enhanced on 5 days after status epilepticus. So, so this model, these results are from the model presented yesterday by Professor Pitcairn. So this is lateral fluid percussion on rats. And here, actually, I don't want to complicate this because we were comparing 2 different machines, actually, to doing multiplex analysis. And this, so the top row is Bioplex and MagPix is the bottom one, but this is not relevant. So what is relevant here is that in the, you have the ipsilateral cortex here, and the next slide is the contralateral. And you can see that CCL2 also was greatly enhanced in the ipsilateral cortex of these LFP rats, and this was detected by both machines, and both were run on triplicates. So this was a very strong indication that CCL2 also can be a very strong marker for traumatic brain injury. This was published in the Frontiers in Molecular Neuroscience in 2011. So this next slide is the contralateral cortex, and you can see that CCL2 also is elevated in contralateral cortex, but you have other cytokines that are very interesting, like IL-18, and you have here TNF-alpha. No, TNF-alpha actually is elevated only on the ipsilateral. This is interesting, actually. TNF-alpha was elevated only on the ipsi and not on the contralateral cortex. So we can say we can be pretty sure that we can affirm that cytokines are altered in pathologies. So, but we don't have still like the mechanistic ways of action of cytokines and which relevance they have in this pathology. We are still trying to figure it out. So we have still to block these cytokines during epileptogenesis or right after traumatic brain injury to be sure in the experimental animals what kind of outcome you can have. So this would be the next step. So keeping track of our questions, I would say that are they relevant in neuropathologies? Well, probably. I would say that we are in the yellow state, probably, but we are not sure if they are relevant, and we are not sure, but they seems to be altered in many, like it's interesting because you have maybe the same cytokines in different injuries to the like traumatic brain injury and status epilepticus, and you have the same, not exactly the same pattern of cytokines altered, but you have many cytokines that are altered in many different pathologies. So it says to me that probably they are secreted in any injury that happens, like fever, and we have this epilepsy. Epilepsy can come from so many different injuries to the central nervous system, and it will be interesting if these are common mechanisms. So these chemokines and these cytokines are a common mechanism from injury to epileptogenesis. But this is a question that needs further research. So this leads us, so, well, if they are relevant in neuropathologies, can they be therapeutical targets? And this will be one of my questions nowadays, and I'll show why, because when I was thinking about, when I have these results with CCL2 alterations in both traumatic brain injury and status epilepticus, I was thinking, how do I block these chemokines? Because you don't have drugs that are antagonists of the receptors, so how is it going to be? And I realized I have to make an infusion of monoclonal antibodies, and I was thinking like, well, but this is a research setup. What are you going to do when you have to move to patients? How are you going to make a clinical trial for monoclonal antibodies? And I was out of touch of how fast things are moving on treatment, on pharmaceuticals. This was quite impressive to me when I realized like things are moving so fast. So what's going on now is that biopharmaceuticals are available for the treatment of rheumatoid arthritis. And you have they have these very interesting names, like I will not try to pronounce. I have no idea how to say this, but they all finish with MIB, you see, so it's monoclonal antibodies. So it's very simple to realize that this is a monoclonal antibody. So you have like these two here for blocking TNF, and you have this one here to block IL-6. So to block IL-6, like I shown you, IL-6 is elevated in epilepsy patients. So to block IL-6 would be our first option to block, even in experimental animals, or maybe for later on for the clinical trial, phase 1, phase 2. But what I was really surprised is these drugs nowadays, they account in Brazil for 40% of the expenses of our national health system. I was really surprised when I realized that. And actually, in the United States nowadays, it accounts for 70 to 80% of the expenses on pharmaceuticals. Because of two phenomena. Because you have the generic pharmaceuticals, so the price for the chemical generated ones are going down. And of course, because the pharmaceutical companies can keep a large difference in price for this, because the US law forbids generic companies to make biopharmaceutical equivalents. So, because of course they are produced in transgenic animals, they are proteins, they have to be purified, and they have to be injected subcutaneously. So of course they are considered brand new drugs. They're dangerous. They still don't have a long-term use of these drugs in patients. So that's why you have to keep track of, you keep it more closely watching. So and this drug here, the etanercept, is a different one. He's a receptor. He's a TNF receptor modified. So it's soluble, like many cytokine receptors actually exist in soluble forms. And this drug here, the etanercept, it combines with TNF and nullifies the action of TNF, because it cannot, it's like an antagonist, competitive antagonist, it works like that. So it can connect to TNF in the cytoplasm and it nullifies its actions. So okay, so thinking about this, so these are expensive drugs. They are making like, I think just one of these is like $10 billion sales in the world. Nowadays, the adalimumab, this is the only, I forgot to say that these are chimeric antibodies. So normally what they do is like they humanize the antibody, that the part that connects with the cytokine is from marine animals, and you have to combine the FC chain, so you have to humanize. And this one is a whole animal, a whole human antibody, so it's considered safer than the other ones. So this is used for rheumatoid arthritis patients and is approved by the Brazilian Drug Administration. And then it comes to our last question. And so I will label this one also yellow, because I think like, can they be therapeutical targets? Well, probably. And it's expensive, but probably they can be therapeutical targets. So this leads to our final question. So is it economically feasible? So if it is so expensive to treat this, why waste time researching this? So I was really glad that Brazil has this huge national institute. So Manguinhos is a big pharmaceutical factory that makes most of the generic drugs for the national health system of Brazil. And now it's just starting this year, the last 2 years, Manguinhos is producing immunobiologics. So it's producing exactly these antibodies to treat rheumatoid arthritis, and they are part of the therapeutical recommendation of the Brazilian government. And they are part of the, was no pun intended, this SUS, is almost save our souls, is our national health system, and people who have to go to a public hospital in Brazil would look at me, marvel, and think like this guy is completely mad because we don't have gauze, we don't have basic things in public hospitals. But it's really strange. Actually, maybe you don't have basic consumables to practice medicine in Brazil, but sometimes you have these very, very advanced drugs to treat cancer, and also these antibodies will be available. So to answer our last question, it looks like, yes, it is economically feasible, even in a country with limited resources like Brazil, to treat people with this kind of antibodies, because in the long run, we're thinking like in economic terms, in the long run, we will save a lot of money because you avoid patients that will have long-term disease, and they can reach like a center like here in Ribeirão, a surgical center where you have to make a amygdala hippocampectomy removal, and it's very expensive one. So if you avoid this kind of development, if you avoid this kind of epileptogenesis to happen and these patients to reach such a surgical level, it will save money for the national health system. So that's what I have to say. Thank you for your attention.